We are going to be discussing the hygienic behaviors of bees as an efficient mechanism of resistance to the chalkbrood disease. To start, let's learn a little bit about the bees. Apis mellifera. The western honeybee is distinguished by their production and storage of honey. Honeybees represent only a small fraction of the 20,000 known species of bees. It is estimated to be only about 1 million years old. The queen has 10 times the lifespan of workers. The queen will lay eggs in a drone or worker cells. The eggs will become fertilized and develop into workers or queens. The unfertilized eggs will normally develop into drones. But to understand what brood is, we first need to understand the adolescence of a bee. The honeybee goes through 3 stages in its life: the larva, the pupal, and the adult. The larval phase is the main period for feeding and growing. The larval phase will end at the beginning of the metamorphosis phase. This phase consists of complex reorganization of larval structures and the formation of immature structures. While still in the larval phase, the bee will start to form the pigmentation in the eyes and the body to prepare for adulthood. But first we must ask the question, is the hygienic behavior of bees an efficient mechanism of resistance to the chalkbrood disease? So what makes a hive hygienic? Hygienic behaviors help to maintain the health of the hive by limiting horizontal transmission of pathogens and population growth of parasites. Chemical signals, genetic predisposition. In colonies, the elimination of brood consists of adult bees removing or cannibalizing the infected or non-normal brood from the individual cells, either in pieces or intact, and moving the cells outside of the hive. The exact origin of chalkbrood disease is likely unknown, but it most likely arrived from Europe when the alfalfa leafcutter bee, when the bee species was introduced to assist the pollination demand in the United States. Chalkbrood disease is caused by the heterophthallic fungus Ascosphaera apis. The fungus spores enter the larval by food and germinate the hind end of the gut around the time when the cells are sealed. This is around 8 or 9 days in the cycle. Mycelia rapidly expands, crosses the heterophthallic membrane, reaches the larval surface 3 days later, and continues their aerial growth. Most of the larvae die when they are stretched from the cells. Larvae killed by the chalkbrood suffer a progressive change in their appearance. If spore cysts are formed as a result of mating, infected larvae will eventually become gray or black in color. Larvae infected by mycelia, but not by strains that have formed spore cysts, are white. There are also treatments for this disease. Study has shown that hives that have removed mummified corpses were less affected by the diseases than those who just left the diseased sections. By freezing the contaminated sections, it keeps the bees who have not been affected from being infected. But how does this disease spread through the colonies? Pollen has shown to be a carrier for these fungal spores, which will lead to a transmission of diseases in the hive. If the diseased brood is not removed from the hive, it will continue to spread and grow throughout the hive by producing more spores. Behavioral resistance is an important mechanism of social control of chalkbrood. In order to test if a hygienic colony is more effective in removing chalkbrood disease, a study was conducted. 31 experimental colonies were tested for this disease, and all came up to be free of the disease. The queen was allowed to lay for 6 to 8 weeks until all stages of brood were present. Pollen cake patties were prepared for pollen pellets collected from mixed floral sources by honeybee colonies by the use of pollen traps. The mixture contained 40% of pollen, 35% sucrose, and 25% brewer's yeast by adding a sufficient water to the pollen pellets to dissolve them, and then adding water to the pollen pellets to dissolve them, and then adding sucrose. This mixture was stirred until no pollen pellets were present. Chalkbrood disease spores were also introduced in the mixture. The first mummies were appeared after 3 days. After 3 weeks, most of the hygienic colonies were removed from the mummies from the combs. The result ended with 7 experimental colonies that were hygienically fit to survive the brood disease. 4 uncapped and removal dead larvae within 24 hours were considered resistant to fungus, and 3 required over 6 days to accomplish this and were termed susceptible. Chalkbrood mummies were not found in 2 colonies that were resistant. A different colony had 2 on the first day of sampling, but there was no others seen during this observation period. The 4th colony had a total of 4 mummies and 3 tests that were taken. The rest of the colonies were filled with mummies. But all 27 of the colonies with mummies showed the same correlation between uncapping and removal of the dead brood and resistance of the Chalkbrood disease. In conclusion, the more hygienic the colony is, the faster it is going to get rid of the disease, like the Chalkbrood fungus. The research that has been conducted has made a path for further research. We have learned that the hygiene of hives can help build up resistance towards diseases and aid in the conservation of bees. We do need to conduct more research on the potential correspondence between the brood stimulus intensity and the level of pathogen infectivity in relation to hygienic detection of the individual and elimination of the infected brood.